Alright, on to our antifungals. Basically, we break it down into two groups. Either it is an infection that is systemic in your blood, bones, or lungs, or dermatophytic, so on your skin, nails, fingernails or toenails. Candida albicans is a major fungal infection that you find on the skin and the mucous membranes, and so we'll kind of like talk about that one a little bit different. But here you can see the different drugs of choice for each type of these infections. With your systemic infections, fluconazole works pretty good. It's in a group just collectively called your azoles. It's a triazole, the third generation. It's a really good antifungal. Amphotericin B is another really good antifungal. Mycophamine is pretty good. Griselfolvin is really good for your dermatophytic infections. And then for your albicans again, fluconazole, urisols showing up again. Nystatin, we'll talk about that one too specifically in a little bit. So with your Candida infections, there are going to be all like superficial infections. They increase with your antibiotic use. You remember when we were talking about superinfections with Candida albicans in the last section in our antibiotics. As you clear out the useful bacteria, Candida takes over and is pretty like opportunistic, parasitic about taking over the new spots. Here you can see a Candida infection in the mouth, back of the throat. This is called thrush. If it happens in the vagina or the penis, it's called a yeast infection. Both of those are fairly common. So our antifungal drugs, we have fungicidal drugs and fungostatic drugs. The fungicidals are like your bactericidals. They're going to kill the fungus versus the static ones, which are just going to stop them from reproducing more. And so your body can get rid of it using your immune system, or with griselfolvin, you just shed it. And we'll talk about that too. So with your fungicidals, a lot of these are going to disrupt the wall synthesis, kind of like we saw in our bacterials. They can inhibit the fungal DNA, again, kind of like we saw in our bacterials. Or they can bind specific proteins that they need for life. Altering the cell permeability, stuff like amphotericin B, nystatin, and the azoles do this. They all affect ergostol, which is kind of like cholesterol, but in a fungus. And so it helps to make up that cell membrane and give it some structure, some rigidity. And without it, you're going to have holes poked into your cell membrane, or it's going to be weaker, easier to rupture, breaking apart, influencing the cell permeability, interfering with the essential proteins. Flucytosine interrupts the RNA synthesis and fungal DNA replication. And then our fungostatic ones, again, griselfolvin, binds keratin, and it binds keratin for about a month, which means that the fungi can't use that keratin. So we're talking like fingernail, toenail infections. Also, the keratin within the skin, right? So ringworm, jock itch, athlete's foot. And so it binds the keratin that is on the surface, stopping the fungi from binding to it. And so it can't spread to new areas. It also binds the growing fungi. It inhibits its mitosis so it can't spread to new areas. And then you just shed off that skin or your toenails or your fingernails, like they grow out within that month, and then you're free of your fungal infection. Really handy. There are a lot of different antifungals out there, so we're just going to kind of keep it to like the major groups. So here you can see kind of where they all work. that just says fungal cell membranes under this picture of me. So here is your fungal cell wall made up of chitin proteins and beta-glucans. And then down here is where your ergostol is, and you can see your amphocytarin B and niacin are going to work here and just poke holes into the cell membrane. Really lets out a lot of potassium, which then influences how the fungal cell can work and dies. You have your echinocannidins, which stop the formation of your beta-glucan synthase, which again is important in making beta-glucans. So messes with the cell wall, it's going to go through a lysis stage and die. Your azoles stop the production of ergostol. Your terbinafines do the same thing, just a little bit earlier, stopping the formation of squalene to squalene epoxide, which then can go on to make ergostol. Your flucytosines work on the DNA, RNA. Your glycofulvin stop it from using the keratin. And so here you can see some of the major ones we talked about, amphocytarin B, and then the non-generic name. Niacin, miconazole, which is you find in monostat. Your terbinafine, which you find in Lamisil. Lotrimin, butanafin, fluconazole, difelcan. And that's it for the fungals. Let's go ahead and start talking about your antivirals. So your viral diseases, these are going to be a virus, it's a small infectious unit made up of either DNA or RNA, depending on your virus. You have things like reverse transcriptase and a lot of stuff we're not going to go into because this isn't a virology class, but just kind of know that you have these packages of DNA surrounded by these protective coats. Some have lipid coats on top of that, or envelopes, so they have kind of like a double covering. Your viruses are really simple genetically. They only carry 1 or 2 enzymes that decode the genetic structure. Really, it's all about, here's my genetic code, put it into the host, and have the host do all the work for me. They're very, very simple organisms, quote-unquote organisms. Are viruses alive? It depends. Because one of the things that is, like, is it alive? It's able to replicate itself? And viruses can't. They have to use a host. The diseases caused by viruses are stuff like influenza, chickenpox, cold sores, mononucleosis, pneumonia, rabies, AIDS, a lot of different ones. And they're going to be very specific for where they attack, like your rhinoviruses and the upper respiratory stuff, eye infections, your parotid glands getting mumps, cytomegalovirus in your pharynx. Like, all these are pretty specific for what they're doing. Rotavirus, coronavirus on the GI tract. We know that coronavirus can also attack the lungs, right? So, the best way to beat a virus is vaccines. Getting the vaccine before you get the virus, your body's already built up the immune defense, and so when the virus enters, you kill it. Easy peasy. There's a lot of viruses out there that we can't make vaccines for because they evolve too fast. Their mutation rate is too high. They're too variable to come up with a vaccine. The flu vaccine, we have flu vaccines. Every winter you should be getting them starting around October, right? And it's to prevent flu outbreaks. About 8 months before scientists isolate different types of flu, typically 3 strains, 2 of strain A. Strain A are kind of the strain of influenza that you get in normal settings, where strain B is stuff that we see in like retirement communities and hospitals where people are really contained within small groups and their immune systems aren't great. This is where you get outbreaks of B. Typically, in normal settings, you're going to get outbreaks of A. Like the swine flu, H1N1 is an A type of flu. The Spanish flu was an A type of flu. And so we use 3 strains, 2 from A, 1 from B, and you grow these viruses in chicken eggs. They grow really well in chicken eggs. That's why if you're allergic to eggs, you probably can't get the vaccine, right? Because it has a lot of albumin in it because they were grown in eggs. When we properly match up the flu vaccine to the strains of flu that are spreading throughout the community, you get about 70 to 90% effectiveness. Pretty good. But people still get the flu. Even if they get the vaccine, there's still a chance of getting the flu. Even if you match it right, there's still a chance of getting the flu. But the flu also mutates, right? So as it's out in the population, you have these things called antigenic drift and antigenic shift. Antigenic drift is kind of like small changes to the outer part of the influenza virus, to where shifts are where you have these big, large changes in these things called hemagglutinin and the neuroaminidase. And these are what the hemagglutinin helps the virus get into your cells, and then it replicates into your cells, and then it bursts out. When it bursts out, the neuroaminidase kind of like cleans off the hemagglutinin so it can bind to another cell. But seriously, like, vaccines are the best way to prevent getting infections. And it's a great way to protect people who can't get the vaccine. So if you get the vaccine, you're not going to be spreading it to somebody else, right? And probably everybody in this group has had one vaccine or another. So if you ever want to talk to me about vaccines, vaccine safety, like, hit me up. In this next group, we will be talking about the different antiviral drugs